Let's move on to our next dairy presentation. We'll have Dr. Sangeeta Rao from Colorado State University, as well as Nora Jean Nealon presenting.
Thank you, Dave, for introducing. As I'm presenting along with one of my students from DBM PhD program who will be graduating next year, Nora Jean. So coming straight to the topic is we're presenting on the antimicrobial resistance from dairy worker clothing and environment, which is an occupational health risk. And it is really important to mention that as well as address that. So we all know that farm workers, including dairy workers, are essential workers who have major contribution towards food security generated from livestock. Workers' health is very important to maintain. And in the light of COVID, we have realized that more than before. We have realized it more than before. And although there are many policies and safe practices, the workers undergo various health risks, including the ones from zoonotic pathogens, and moreover from antimicrobial resistant pathogens. So this is what our study is about. And this is a follow-up of a project that was performed by one of our former graduate students, Dr. Palomares, who graduated a couple of years ago. And it was to assess the socio-ecological factors among the dairy worker population to zoonotic pathogens. So this study, what we have done is a follow-up of the study that was performed a couple of years ago, and the sampling was performed a couple of years ago, and we conducted antimicrobial resistance study on those. So zoonotic pathogens could implicate high risk to the dairy workers who are in close proximity to these animals, and we all understand that. And these animals who could be shedders of these pathogens, especially Salmonella enterica is the one we focused on, and especially Salmonella Dublin. And the AMR or the antimicrobial resistance composition of those pathogens that have been isolated. So risk could also be to the family members of these people who are in close contact with these workers in their daily lives. So these workers can bring those infectious pathogens home, which carry antimicrobial resistance. So we all know that emerging and existing antimicrobial resistance is a major public health concern, especially from these foodborne pathogens and food-producing animals. And when somebody gets affected, treatments become more challenging, and it is important to address this issue. Of course, there are other confounders such as behaviors and practices of these workers, which play a major role in the risk of exposures to these pathogens during any of these activities. And worker clothing could be used as a proxy to assess these risks of exposure to these enteric pathogens. Hence, it's important and essential to understand the distribution and diversity of AMR across Salmonella enterica genome in farm worker environments, and to link to specific epidemiological sources to equip us better with mitigation of AMR. When I say AMR is the antimicrobial resistance, I should have mentioned that. So when I say epidemiological sources, what we did was we collected samples from the clothing of these people, which I will come to the method and I'll explain more. And we have used whole genome sequencing as a tool to be able to identify this genetic material, which carry antimicrobial resistance. And whole genome sequencing is a comprehensive method, which provides information on the genotypic characteristics of these pathogens, including the AMR determinants. And agencies like CDC, they use whole genome sequencing to be able to find out any foodborne pathogens, to be able to find out the sources of these pathogens. So we have used a similar technique to be able to identify the antimicrobial resistant determinants. So the main objectives of our study were to identify and understand the epidemiology of antimicrobial resistance of Salmonella enterica, especially Salmonella Dublin, isolated from dairy worker clothing and environment, and to determine the genetic diversity of antimicrobial resistance. So workers were selected within the milk parlors, calf rearing areas, maternity and hospital areas. And the samples from milking room were also collected as an environmental sample representation. And one of the limitations in this study is the sample size, as we were not able to collect a lot of samples, and it was only one farm that was represented in this study, which does not talk about other farms, what is going on. Maybe that could happen in future. So before the start of the work shift, 3 pieces of absorbent material, like cloth material, was put as a chest piece on the hands, and the boot swabs were also collected. So when the piece of material was put on the hands, also the glove sample was collected. And then differences among these were being evaluated as a source of risk to these people, how close to the face. And in the COVID-19 situation, we've seen how important it is from hand to face kind of factor to be considered. So the potentially contaminated materials were collected after 15 to 20 minutes of their work shift, giving enough time for any pathogen in the environment to be stuck to the material. So all the samples were immediately put into sterile buffered peptone water and transported to the laboratory for further analysis. So we divided this method into 3 stages: sample collection and pre-enrichment, which happens in the farm, Salmonella isolation, and DNA extraction and whole genome sequencing. Whole genome sequencing happened in South Dakota University by one of our collaborators. And then the sequences were sent back to us, where we used a Genius program, which is a very good tool to be able to compile these sequences and blast it against antimicrobial resistance genetic database to be able to find these genes with a particular cutoff. So for results and a discussion, I will hand over to Dr. Nealon, where she will present the results of the whole genome sequences and what we found. Over to Dr. Nealon. Thank you.
Thank you, Sangeeta. So hello, everyone. So following these sample collection methods, 2 farms participated in our study, and these were both sampled during the 2016 year. One was a conventional farm and one was an organic farm. And collectively from these 2 farms, 42 workers were, or 42 workers were sampled, which included bib, boot, and glove samples as previously discussed. And 11 samples were collected from that dairy environment, including different hospital rooms, milking parlors, and pens where calves were. And from these total samples, as illustrated with our infographic on the bottom, 26 samples were PCR positive for Salmonella Dublin, the majority of which we could see were worker clothing samples. And moving on to our next slide, we could see, looking at this pie chart, which represents the results of our whole genome sequencing for antimicrobial resistance genes from these PCR positive samples. We could see across all samples, there were 137 distinct antimicrobial resistance genes spanning 13 antimicrobial classes. Among these, a large portion of this pie chart were beta-lactam resistance genes, which included classes A, B, and C beta-lactamases, penicillin binding proteins, and outer membrane efflux pumps for different beta-lactam drugs. And from a public health perspective, this can help confer resistance to many beta-lactams used conventionally, both in dairy cattle and people, including a few used here, which are used for different uses in both dry cow and lactating cow therapies on dairy farms. Also of interest were identification of phosphom resistance genes or phosphomycin resistance genes, which are identified in 3 different salmonella isolates, all from dairy worker clothing. This isn't an antibiotic conventionally used in dairy cattle operations, and it's conventionally a bigger gun antibiotic in human medicine, especially when in human infections or resistance to other antibiotics, which is sort of highlighting a maybe new exposure risk for us that we weren't before aware of. And I wanted to illustrate a 3rd resistance class here, partly because it's one of our whole genome sequencing and database like catch-all sort of gene class. And by multi-drug resistance, these genes refer to different efflux pumps and regulators of efflux pumps that help sort of excrete different classes of antimicrobial drugs from bacteria, many of these genes more than 3 different classes at once, which could alone confer multi-drug resistance to these Salmonella isolates. And moving on to our next slide. So what I did here is from these antimicrobial resistance genes that were identified, I now grouped them by their isolation source. So they could be 3 different clothing types from the workers and worker samples, which are in orange, or blue, which represents environmental sources. And when we look at these worker clothing samples in orange, we tend to see overall, when comparing these total number of genes to environmental genes, there were higher numbers of antimicrobial resistance genes isolated from workers versus the environment. This is particularly true looking at this green arrow for beta-lactam genes, and when we compared to the lower numbers of beta-lactam genes isolated from Salmonella environmental samples. And then, interestingly enough, with this second lighter green arrow, phosphomycin resistance genes were identified only from worker clothing and not in environmental samples, and the implications of this were worker exposure and where these genes might be coming from are still being investigated, including both with genome sequencing as well as with some antibiogram evaluation, which is ongoing. As we move on to our conclusions and next steps for this analysis, overall we could conclude from our preliminary isolation that Salmonella delphine is prevalent on dairy worker clothing and the dairy worker environment. This is a known exposure risk for dairy workers, where Salmonella delphine is a common pathogen encountered in dairy cattle, both in like the dairy environment and within the dairy cow as workers work with them. And the beta-lactam resistant, I mean, it's known that a lot of gram-negative enteric bacteria, such as Salmonella, are emerging with antibiotic resistance and pretty escalating levels to beta-lactam drugs. This continues to be a public health concern for the CDCs, especially with the growing rise of ESBL resistance among Enterobacteriaceae microbes. However, phosphomycin isn't conventionally used on dairy farms, and this warrants further investigation into like its exposure risks and maybe potentially as an emerging exposure risk in livestock production. There have been some previous studies that have identified this in broiler operations and swine operations, but limited evidence to support that it's a current risk within dairy cattle operations. And as we move forward to expand on these results, we are performing antimicrobial susceptibility testing of both worker clothing and environmentally isolated Salmonella samples to compare along with their whole genome sequencing resistance gene identifications to see what they may be harboring versus phenotypically expressing. In addition, because Salmonella delphine has potential to be a pathogen, especially for people as a zoonotic agent, we are additionally looking at the virulence factors through whole genome sequencing that these Salmonella isolates may be harboring. And an additional sort of detailed study that Dr. Rao had highlighted briefly with our methods is these Salmonella samples, when they were initially swabbed and taken to the lab to be cultured, they were grown in 2 different media types, RV versus TET, which are 2 common ones that we grow Salmonella in for enrichment. And the sort of use of these different medias and how that might influence what we're identifying downstream in gene identification is also being explored to ultimately sort of inform on like methodology and best practices for like workflows in identifying these genes. And with that, we leave with the questions of what do different frequencies of these genes from worker clothing versus the environment suggest about these exposure risks? And then second, what are some phenotypic versus genotypic resistance patterns, especially for beta-lactam and phosphomycin resistance genes, which represent a known and potentially emerging exposure risk in dairy farm workers? And with that, we'd like to thank our many collaborators and sources of funding. And thank you.
Thank you all for your attention. Any questions, we'll be happy to answer after the session.